second.
Hello, Assalamu alaikum. I am Dr. Jamil from the Physiology department, and this lecture is in continuation with the renal physiology. And today we are going to discuss a very important university question, that is the reabsorption of the glucose, how the glucose is going to be reabsorbed. So the glucose reabsorption occurs, as I have told you earlier, that only in the PCT. Any glucose which remains unabsorbed from the PCT and enters into the lumen of the nephron beyond the PCT, that means the distal convoluted tubule or loop of Henle or this and that, that will not going to be reabsorbed. So all of the glucose is reabsorbed within your PCT. So glucose reabsorption is the secondary active transport because it is sodium dependent, and from the luminal surface 90% through the sodium glucose transported to in the initial part of the proximal convoluted tubule, and remaining 10% is reabsorbed by the sodium glucose transporter in the remaining part of the proximal convoluted tubule. So glucose reabsorption is secondary active because it is going to be reabsorbed along with the sodium. Then on the basolateral side, glucose diffuses out of the cell by the glucose transporter 2 in the segment 1, S1 segment, and glucose transporter 1 in the last part of the PCT. All the glucose up to Tm is reabsorbed in the PCT, as I have told you. The Tm definition I have told you earlier, the maximum rate at which the tubule can reabsorb a substance per minute is known as the Tm for that substance. Now for glucose, the Tm is 375 mg per deciliter. That means if your glucose level is near 375 mg per deciliter, even then all of it should be reabsorbed. Now we have got another definition which is known as the threshold of the glucose. It is the minimum plasma level of the glucose at which it begins to appear in the urine, and it is just 200 mg per deciliter. Now this threshold means the amount of a substance in the plasma when it is being appear or become secreted in the excreted in the urine, and this is 200 mg. These two statements, that is that according to the Tm, it should be reabsorbed up to 375 mg, but according to the threshold, it appears before that Tm, it appears just at the level of 200 mg per deciliter. So these two statements are contradictory. These two statements are not matching with each other. If we consider only Tm, it should be reabsorbed up to 375. If we consider threshold, it begins to appear just above 200 mg per deciliter. And why this contradiction is there? Why these two statements are opposite to each other? Although these two statements are correct, both of these are correct. The Tm is 375, in reality it is so, and threshold is 200 mg, again it is so. These two statements remains there because of a phenomena which is known as the splay of the glucose. Splay of the glucose. Not phenomena of display. Sorry, my laptop was not following my commands. Now, before that, before discussing the phenomena of display, let's understand some figures. That is at the GFR, which is 125 ml per minute, Glucose concentration is 250 milligram as explained below. So glucose concentration at the level of GFR is 250 milligram. This is a normal phenomenon. At threshold, it is 200 milligram per deciliter. 200 milligram means from where we have got, actually we want to know from where we have got the figure of the 250. We want to justify this 250 milligram. Now at threshold is 200 milligram per deciliter. 100 ml, that means 100 ml contains glucose 200 milligram. If we consider this one at threshold 200 milligram per deciliter, deci means 100. So 100 ml contains 200 milligram. Now 1 ml contains 200 milligram over 100 and 125, which is the GFR. At the GFR, at 125 we will do the formula, we will use the formula 200 over 100 multiplied by the 125, that is 250 milligram per deciliter. So the filtrate contains 250 milligram of glucose, and this is less than the TM, that is 375 milligram. So the glucose should not appear in the urine at the threshold of 200 milligram, but it do appear due to the splay phenomena. Why the splay is there? What is meant by the splay? And we are going to understand this thing with the help of a graph. These are just figures, just to make you understand that glucose should not appear at the level of, these figures are not necessary to be memorized. Just again try to understand one statement, that Tm is 375, threshold is 200. These two statements are not matching with each other. At the 200 milligram, glucose should not appear in the urine because still we have got the carrier proteins, still we have got the capacity in the PCT to reabsorb the glucose. Now, why this play is going to occur? Here are the two graphs. First of all, what is meant by the splay? Splay means to move outward. Splay means to convert an acute turn into the rounded one. Splay means to extend your feathers, extend your arms, or to make it a rounded. This is a technical of splay. So this is a graph in which you will see this is a red line. This is the red line is concerned the glucose reabsorption, and this red line is actually the filtrate which is coming amount, which is coming in your nephron. This is the filtrate coming in, and this black line, this black dotted line is the concern with the reabsorption of glucose. So if you see that according to TM 375, this is your TM where my cursor is, the glucose which is filtered out, it is reabsorbed. So the filtration of the glucose and the reabsorption, they are coinciding with each other. So amount of glucose which is filtered out, it is going to be reabsorbed up to the TM. And these two lines, they are these single continuous red line is going to coincide with the dots which are concerned with the reabsorption of the glucose, because according to the theory of the TM, all the filtered glucose up to the level of the TM should be reabsorbed. So beyond the 375 milligram, the reabsorption will not be there, and this line is going to be straight now. It's going to be straight now. Reabsorption will not be the further one. On this side, we have got the movement of the glucose in milligram per deciliter, and on this side, the renal threshold level. So we have got an acute turn over here. Up to 375 milligram, they're straightforward, they are moving with each other, and then their pathways are different. Filtered line is moving above, and reabsorption line is going to change its pathway. Now let's see another line, which is the green dotted line. This is the green line, broken line. This is concerned with the excretion. So if we consider the TM, then the glucose should be excreted or should appear in the urine according to the threshold. It should be appear approximately again at the level of the 300 or beyond that one. So the glucose excretion should begins to appear from this line. So this graph is according to the threshold. This graph is according to basically TM, and according to that TM the threshold should be like this graph. Let's see another graph. Here is another graph. that again your red line which is concerned with the filtered load of the glucose, and we have got the blue line which is concerned with the glucose reabsorption. And sorry, now you see that here this is your approximately, if you follow my cursor, this is the approximately 375 milligram. But before that 375, before that, the glucose reabsorption line is going to make a splay. It is making outward turn. It's make a turn in this way, and then it's going to be state. Before that level of the TM, it is going to make it. These two lines are going to separate it. This turning of the two lines, this turning of the blue line, making a rounded circle or rounded line. This is known as splay. This is technically known as splay line. It's just like when you're reaching when you're driving and reaching a road which are connected with in a T shape. So you have got a slip road to approach the next road. So these slip roads are also the splay. So this is turn is the display. So the glucose reabsorption is not following the TM actually. The TM line, it should be 375, but actually it is going to take a turn before that one. And here you will see in the excretion line. In the excretion line, in the previous diagram, it was starting from this point up around about 300. But this is going to start in reality from the threshold level. That is the 200 milligram per deciliter. Again, that previous straight line is not going to have a turn. It makes a turn over here. And this is moving in this direction. This is again a splay. So this phenomena of splay is that the splay, why the splay is there? Because you conceive in your mind that your bladder is, your urinary bladder receiving the urine which is formed by all the millions of the nephrons which are present in your both kidneys. And once they are collecting, your storehouse of the bladder is receiving the product of millions of the nephrons which are present above in your kidneys. And all the nephrons, they are not functioning up to the mark. All the nephrons, they are not functioning equally. That's why some nephrons, they begin to, their reabsorption is not up to the level of the TM, and that threshold starts at the level of the 200 milligram, because all the nephrons, they are not functioning equally. Although the nephron, a good nephron should work according to the TM, but some of the nephrons, they don't follow the rules and they don't function up to the TM. Their function is suboptimal, less than TM. So the glucose begins to appear in the urine just at the level of 200 milligram per deciliter. So this graph is showing both the phenomena, that is the splay of the glucose having a turn on this one. So this was all about the glucose reabsorption. Here is the diagram. You can understand that sodium glucose transporters, sodium, this is the sodium and this is the sodium carrier protein, along with the glucose they will move enter, and from here the glucose will move through the glucose transporter 1 and 2 to the interstitial tissue spaces. And this is the concern to the previous thing. These are brush borders. So this again I am going to repeat that the TM and the threshold level both are correct, but these two are not matching with each other. And this you can say not equal, you can say these two statements are not matching with each other, because all the nephrons, they are not functioning up to the mark, they are not functioning up to the standard level. Some of the nephrons, they do not follow the TM, they do not work the TM up to the TM, and glucose begins to appear in the urine. Thank you very much. In the next lecture, we will move, in next discussion, we will move to the reabsorption of further few substances. Thank you very much.